Okay, so today we have a pleasure to have Professor Jens Meiler in this bioinformatics seminar series. Professor Meiler got bachelor's degree from Leipzig University in Germany, and then after that he got PhD in University of Frankfurt. Then he came to the US at Professor David Baker's lab as a postdoc, and he spent there from 2001 to 2004. As you probably many of you know, the David Baker's lab is one of the most influential and successful lab in protein structure prediction and protein design. And then after finishing the postdoc, Professor Meiler moved to Vanderbilt University, Nashville, and where he is currently a distinguished professor in chemistry, pharmacology, and biomedical informatics. Then also he has another position now in Leipzig, back in his town in Germany. And I just heard that he is flying back and forth between two labs, maintaining two labs. So he has over 215 publications. And I see that Professor Meiler has been very successful in structure prediction, but also applying the structure modeling to various different biological problems. And since now we see that the protein structure prediction has improved a lot in the past couple of years, this is really a good time to hear from Professor Meiler how we can use that computation model in various biological real problems. So, okay, thank you again for Professor Meiler to join us. Okay, please, maybe you can share the screen and go ahead in your presentation. Okay, so yeah, his talk is on computational structure prediction and therapeutic design with Rosetta. Professor Meiler, please.
Okay, so I hope everybody can see the screen and you guys can hear me well. Excellent. Yeah, thanks again for the invitation. It's a great pleasure to visit virtually, at least, and talk to you a little bit. What I've prepared is sort of an overview of some of the research that we are doing and some of the more recent developments. There are a lot of topics in here. Some of them I will go in a little bit more details. Others we will just skim on the surface. But I'm obviously happy to answer any questions as they come up. So my laboratory really works in three major areas, which were already mentioned. The first one is protein structure prediction. Specifically, we have an interest in membrane proteins, and specifically there we look into mutations in membrane proteins that cause human diseases. And then the second area of the lab is what I would call small molecule or traditional drug discovery. Here we really look for small molecules that often also bind to these membrane proteins. So we build the models on the left-hand side, and then let me change the pointer here too, so that's more easier to say. We build the models in this first portion of the lab, and then we use them for structure-based drug discovery in the second part. We also employ a lot of machine learning, artificial neural networks, artificial intelligence, if you want, in drug discovery and also in protein structure prediction and in protein design for that matter. And the third area of the lab is engineering proteins, engineering antibodies, engineering protein therapeutics, engineering vaccines. And I'm going to show you a few things of each of those. Before I get into that, I wanted to just formulate the problem again. Given the amino acid sequence, what's the three-dimensional structure? And we all know why this is important, right? Because the central dogma of structural biology is that sequence informs structure, informs function. So if I understand the structure in the center, then I can better relate the sequence to the function of a protein. if I have a small protein with 100 amino acids, each amino acid has maybe 100 conformations. That's 100 to the power of 100 possible conformations. That's 10 to the 200. That's a lot. If a chemical bond can rotate, with a maximum frequency of 10 to the 14 per second, then you still need 10 to the power of 186 seconds to sample all of these conformations. That's longer than planet Earth exists. And that was formulated way back in the 60s. Several people had similar formulations. The best known is Leventhal's paradox of protein folding. Nature cannot fold proteins by sampling the whole conformational space. And we know nature doesn't need to do that. Nature wants to minimize free energy, and so there is a clear pathway to finding the biologically relevant conformations, which is following this pathway of lowering the energy. But it also means if you want to program a computer to solve this problem, you don't want to do an exhaustive search. You want to be somewhat smarter. And 20 years ago or so, while I was in the Baker lab doing my postdoc, this algorithm was developed called Rosetta. And here we see a folding simulation. That's about real time on your PC. Without going into too much detail, it's a Monte Carlo algorithm. So it applies non-conformational changes, predicts the energy before and after, and typically accepts the conformational change if the predicted energy is reduced. And if the energy goes up, there is a probability criterion, a metropolis criterion. Mostly these steps are rejected. Sometimes they are accepted so that we can leave a local energetic minimum. And this is ubiquitin, 76 amino acids. And you see that this algorithm ends in this particular trajectory in a conformation that's pretty close. Takes about 36 seconds for a small protein. For a large protein, it can take 20 minutes or so to fold. It's a stochastic process. You don't run it once, you run it thousands of times, 10,000 times, sometimes 100,000 times to sample the conformational space of a protein completely. Now, you do know, or I tell you now, that Rosetta not only folds proteins, Rosetta really developed into a platform to do almost anything you want to a protein. So that goes to protein folding, but also protein docking. Small molecule drug discovery is the module that I programmed when I was a postdoc, looking at peptides on surfaces of metals, for example, modeling symmetric proteins, and also on designing proteins, designing antibodies, working with non-canonical amino acids, working with DNA. All of these parts are a part of Rosetta. It's an academic software package. It's freely available. You can go online, click, get a license, an academic license, download it, and try to use it. It's developed by 70 laboratories worldwide. There are more than 50,000 licenses worldwide, academic licenses of the software. There are several user interfaces available, but they are not as comfortable as you might know from Schrödinger. This is an academic software. There are 500 people programming on this every day. It's changing constantly. It's a moving target. It's not that easy to learn. So if you get excited about some of those things that I'm showing you today, there is a certain learning curve associated within. My laboratory runs twice a year a workshop where you can learn the protein structure prediction and the protein design basics of Rosetta. If you are interested in that, let me know and I put you in contact with the person who organizes that. But it's a great, it's a great scientific experiment also socially because of this large group of people. And in non-COVID times, we have our joint conferences. This is a few years back, obviously, as you can imagine. The development of the software is focused in the United States, but there are also several laboratories in Europe and then even in China. With that, protein structure prediction, Rosetta was big in the news 20 years ago when it started to really outperform the algorithms that were running at that time. Now, you probably, or many of you might not remember that 20 years ago when Rosetta made it through the news, but you might remember last year when AlphaFold made it to the news with a much improved performance of protein structure prediction. And so you know that we are, these development go in bursts, and Rosetta was certainly one of those bursts. Now, I would say 10 years ago, we had contact prediction, which really made a second burst. And now we have all of these machine learning algorithms which make another burst, improving these algorithms. AlphaFold obviously did a great job in doing that. And luckily now the data are also published and the algorithms, so we can all analyze and understand what's going on. And we are still in the process of doing that. Just wanted to say that also Rosetta has these machine learning algorithms implemented, and they're constantly improving, and people are working on those. And so there is definitely a lot of research also recently coming out of the Rosetta community, learning how to use these machine learning algorithms for these type of tasks. I'm gonna jump through some projects just to try and stay focused and not overwhelm you with too many things. I mentioned we are working on membrane protein structure prediction, and I skipped that. Here's a multi-drug transporter that we did. What I want to talk about is this project here, which is one that I find very exciting in terms of the application. So we call this personalized structural biology, if you like, because you're aware. So why is AlphaFold suddenly so good, right? I mean, the machine learning technology is one thing. The other thing is the enormous amount of protein structures, right? So the protein folding problem isn't solved. Nobody can predict the structure from the sequence alone. They can predict the structure from a multiple sequence alignment and thousands of example structures, right? So there's a lot of data that goes into that. But this large amount of data allows us to predict protein structures better and better. For about 75% of the human genome, you can build a very accurate model for the portion of the human genome that folds in the protein, so the proteome, if you like. And at the same time, the sequence world with next generation sequencing and other things is so rapidly growing. It is not difficult anymore for a patient to sequence the genome and find out what are the mutations that this patient has, that distinguish him from maybe his parents or his siblings that are healthy. And so the idea in this personalized structural biology is, let's bring this together, the really increased accuracy of structure prediction and this knowledge of mutations that might cause the disease in a patient. And by building an actual model of not just the wild type protein, but the specific mutation, I can understand the mechanism, I can understand the function. That's going back to the central dogma of structural biology, sequence to structure to function. And possibly, I can even help to suggest the therapy. I might be able to in the easiest case, find out which approved FDA drug might be the most efficient on the mutation that is on a mutant protein that the patient has. And if this is a variant of unknown significance, if it's a mutation that has not been observed before, we might not know beforehand which is the best therapy to go with. So we build a pipeline so that you can put in your protein and your mutation, and it outputs a model and maps all other known mutations and so on. This is about to be published. What I want to show you is this can be applied, for example, in cancer. So in cancer, we often have variants of unknown significance, new mutations in the cancer cell that have not been observed before. So back in 2014 or whenever we did the discovery, we found this lung cancer patient here in the cancer center. And he was a non-smoker, never smoker. None of the standard therapies really worked. And once they sequenced some of the proteins in his cancer cells, they found this duplication of a domain in EGFR. So EGFR is a receptor tyrosine kinase. When a growth hormone binds, it dimerizes on the extracellular side. And then on the intracellular side, there are two kinase domains. One activates the other, and that sends out a signal. These kinases are often activated in cancers by mutations, but this mechanism where this kinase domain is duplicated was not known at the time. And so we built a structural model for this novel mutation and showed that this red linker between the two kinase domains is long enough so that one can activate the other. So without dimerization, this is always on. This is always signaling, as if the hormone would be there, the growth hormone. And so then the logical hypothesis is this patient should be treated with an inhibitor for that particular kinase, which is afatinib in this particular case. And so the patient was then put on afatinib, which was not the standard therapy at the time. And it's hard to see for, I'm not an MD, but this is the tumor, and you see it shrinking after the first 2 cycles of afatinib. Unfortunately, a lot of this personalized analysis and therapy is nowadays only done, in very late stage patients where all the traditional sort of standard of care strategies failed. And so this cancer pretty quickly acquired additional mutations and started to grow again. So unfortunately, this particular patient died, but this observation gives us hope for the future that if we can begin with a personalized, really molecular understanding of the cancer, that we can develop better treatment strategies for cancer patients in the future. And here's another story. I go back to that. This is just another example here, without going into too much detail, but this is a program called Undiagnosed Disease Network, where patients, often children, who clearly have a phenotype, they are sick, but it's unclear what causes. So there's no diagnosis, undiagnosed disease network. So you take the genome of the child and you also sequence parents and healthy siblings. And by comparing the genomes of parents and healthy siblings with the child, you can identify a short list of mutations, 20 to 50 usually, in proteins that are private to the child, that healthy relatives don't have. And then we build models for each of those mutations and try to understand, is this mutation likely to interfere with the function of the protein? Is interference of the function with the function of the protein likely to cause the the phenotype that is observed in the patient? And if you can get this understanding, then you're often, there might be a therapy available, or at least you have a diagnosis and you can work towards a therapy. So these are some of the structure prediction questions we are asking. Let me go on to computer-aided drug discovery. And this is, again, a field that undergoes a revolution right now. And I believe that this will be at the interface of structure-based and ligand-based drug discovery. So on the left-hand side, we see what I would call structure-based drug discovery. Structure-based just means you have a structure of your target protein and you actually place a small molecule in there somehow. So this is Rosetta, And we see it designing small molecules in a fully flexible protein binding pocket. That's HIV protease. And so this is something that is Rosetta's strength to design into flexible protein binding pockets. Now, for small molecule drug discovery, for many years, since the 70s, since the first computers were around, people built quantitative structure activity relation models. So in the absence of the structure of the protein, they still know the structure of the small molecule. A chemist synthesized 50 molecules, and some biochemist or biologist measured an activity on a certain target in a certain assay. And then the question becomes, how does the chemical structure relate to the biological activity? It was one of the first applications of computers. As soon as they became available, people got excited about this. So this field is very old. And people used machine learning very early in that field. So neural networks in the 90s, as far back in the 90s, there's a book, Neural Networks for Chemists. I think you find still old copies on Amazon if you're looking. That people started to use machine learning for these kind of tasks, for this classification task, and it really turned out that machine learning is very powerful. So now we have multiple developments here. The first development is we can integrate these machine learning algorithms with the structure-based algorithms. So while you design your small molecule in the binding pocket, you can run machine learning algorithms that tell you, is this the right molecule for this type of binding pocket? What is the predicted binding affinity? But also, how does this molecule compare to all other known molecules, and is it likely to be active? And so you have this integration. And obviously, with more and more structures for targets and better machine learning algorithms, there is a revolution at this frontier. Another revolution that's happening is on the medicinal chemistry side. In academia in particular, it's, medicinal chemistry is very expensive. You need many chemists synthesizing molecules in hoods. And so that's expensive lab space, expensive personnel. And often a lot of this is done, in companies. In the molecular library program, NIH, 15 years ago also invested massively in academic drug discovery. But more recently, companies came up. They offer make-on-demand libraries. So they have huge libraries, 20 billion compounds or so, but they don't have them physically in hand. They just say, we believe that we have the chemical reactions to make those compounds. But in a computer, I don't mind if I don't have them physically in hand, right? I can take the 20 billion and screen them with a fast neural network approach, and then take a subset and dock them and design them in a structure-based approach and come up with a set of molecules that I'm excited about. And I send them 100 and they try to synthesize them and they get 80 back to you. And then you are in a position where with relatively little resources, you can really jumpstart a drug discovery program, can get a hit compound that has the activity you are interested in on the target you are interested in. And you have a structural understanding how this molecule binds and why it has this activity. And this is really something that's just starting up, but the first papers come out, and this looks like a really promising directory and direction to go into. And so I think also this field is, moving forward with wonderful large steps, and we are implementing in Rosetta algorithms, targeted algorithms that can screen these billions of molecules and find you the one that, or not the one, usually you order 100 and then you get 80 with an 80% success rate. And then you're happy if 5 or 6 of those have the activity, right? This doesn't work perfect, right? But at least you can get a few molecules that have the activity you're interested in. So in this vein, we are actually writing a computer game. Some of you have heard of Folded. a computer game where you can fold proteins and design proteins, tries to engage citizen scientists. And so we are actually working on a small molecule design version of Folded. And the way it's programmed, you can only design molecules that are in the space of 20 billion that I can go out and purchase. And so we have a relatively economic way. If a player designs a molecule we are excited about, there's a relatively economic way to purchase this and test this molecule, right? And so that's something that's forthcoming. There's actually, this is an older video, but just on November 1 on the Folded channel, there was a video introducing this new mode. So Folded is also free. You can download it and help scientists solve problems that are unsolved. I'm gonna again just pick maybe a couple of projects here. We're gonna skip this one, but I show this one for just a second. So in my lab, I don't show a whole lot of method development, but that's actually what I'm passionate about. So most people in my lab write new algorithms. And so just to give you a little bit of an idea of what this looks like. So here on the right-hand side, we see this QSAR idea, chemical structure in, then you translate your chemical structure in descriptors and numbers, and then you train a machine learning algorithm, like a neural network, to output biological activity. And so that's the QSAR model. Mostly the descriptors, these numbers that describe the molecules, are distances and angles, because they are translation and rotation invariant. So if you have a three-dimensional structure of your small molecule, the distance and the angle don't change if you change the Cartesian coordinates. But mirror and image and mirror image are identical when you just look at distances and angles. But in biological activity, we know that mirror image and mirror image can have very different activities. So we want to encode stereochemistry of small molecules in a translation, rotation invariant way that a neural network can understand. So the chemists amongst you will know, the chemists have these complicated Kahn-Ingold-Prelog rules, the SIP rules, to determine if a carbon atom is R or S. And that sort of says, you know, it looks one way, it's the left hand or the right hand, right? But this is silly for a neural network to learn, because R and S, you know, changes with changing a fluorine to a chlorine. You could have a change from R to S. So it's not really useful information. So we developed this you know, descriptor that takes 3 atoms and the center of mass and determines from the shape of all of the triangles between 3 atoms, all the triplets of atoms in a molecule. It determines a volume that is signed. And the larger the volume, opposite sign, you know, defines sort of the largest triangles in your hand. Are they left or right from the center of mass? And that's something that is a geometric parameter of stereochemistry that a neural network can understand. And then we can use these type of descriptors to train our machine learning algorithms. And here's an example for a receptor in the brain, metabotropic glutamate receptor, which is a target for a putative target for anxiety and addiction. And we did an experimental high throughput screen. There were several 100,000 inactive compounds, several hundred active compounds. Here are some of the actives. And then we trained a neural network to learn what distinguishes these few active molecules from all of the inactive ones. And with the neural network, we screened one of those large libraries we can purchase compounds from and bought molecules that are chemically very different from the known actives. So we wanted to scaffold hop. We wanted to have new chemotypes, new chemical structures, but with the same activity. And you see our structures that we purchased recapitulate some structure features of active molecules, but reassemble them in really new and creative ways. And some of those were quite potent, 75 nanomolar. And then we basically use those as lead compounds for the drug discovery program, optimized them, tested them in animals, and proved that this is indeed a target that could be interesting for anxiety or addiction models. Okay, and then let me skip some of the modeling of GPCRs. And here we see some of this merging of protein structure prediction, of structure-based and ligand-based drug discovery. So in this particular case, here on the right-hand side, often we have a situation where we have a good structural model of a protein that we can construct, and we have a library of ligands where we know the activity. The docking program usually takes one protein and one small molecule and tries to dock it. So what this algorithm does, it takes the known molecules, superimposes them, sort of assuming that they have the same pharmacophore, they bind in the same way. And then it docks the superimposed ensemble into the protein, and it asks, find a binding pose where all of these molecules that we know are active truly fit into the protein, and where the relative binding affinity of these molecules fits what we know from our experimental data. And with this approach, we can basically reduce the false positives. So this plot is very small, but on the y-axis here is quality of the model. 0 RMSD would be perfect. We usually want to be below 2 angstrom or so for ligand docking to be happy. And then the cyan on the right-hand side, that's the distribution of models if I don't use the structure-activity relation, if I dock each molecule individually. The left-hand side is if I dock them simultaneously and require that they all dock. And you see a lot of those high RMSD models are not there anymore. They are excluded because one of these molecules is going to clash and just won't fit in this. And we have a much, much higher percentage of low RMSD models that are sampled. Okay, and that brings me to the last 10 minutes or so of my talk, which goes into protein engineering. So now we switch gears entirely. We are not talking about small molecule drugs. We're talking about protein drugs, antibody drugs, and also vaccines. And again, I'm gonna try and highlight some of these strengths that the computational algorithms bring to that field. So the protein design problem goes the other way around, right? We discussed about sequence to structure to function. Now that's structure, that's prediction, and then I know the function. For example, what is the mutation in the patient does. Now I want to go the other way around. I want a certain function. I have an idea how the structure needs to look like. What's the sequence that gives me that function? So I'm basically inverting this. So given a protein structure that I assume has a certain function, what is the sequence that falls into that structure? And again, that's a very large computational problem. This is a very old video from 2003 or so, when the first Rosetta design paper was published. It's again a Monte Carlo algorithm that now I should only show the core of the protein to make it not too confusing, but it samples conformation and sequence simultaneously. So you try all 20 amino acids in each position in many different conformations, and you try to find the lowest free energy state of this. These are some steps that are rejected with very polar cores with a lot of oxygens and nitrogens. But if you only plot the steps that are accepted in this protocol with low energy, you get a very apolar core, as you would expect. And you get a sequence and a conformation that's very similar to in gray, the experimental structure for this protein. This protein is computationally engineered. It doesn't exist in nature. The structure was obtained afterwards. The DNA was put in E. coli and they made the protein and then was crystallized. But this is not a protein encoded in the DNA in any organism. Even this particular fold with the 5 beta strands and the 2 alpha helices in this arrangement was not known to exist in nature at the time. I believe, meanwhile, there are some examples for that fold. And so this protein doesn't do anything. That's a rock. It just folds. But now let's talk about function, right? And so again, what we do is we look into developing cool new algorithms to do that. And I just wanted to mention one here, which is a multi-state design algorithm. So a multi-state design algorithm tries to design one sequence that falls into one structure that stabilizes multiple states. So for example, to make that understandable, if I want an antibody that neutralizes all types of the HI virus, I need one antibody that binds to the surface protein that is on the surface of the HI virus, but not just to one specific HI virus, but through a very large panel of HI viruses and mutants thereof that sort of circulate around the world. And experimentally, that's a difficult problem to tackle, right? But in the computer, you can do that. You can have 35 copies of the surface protein of HIV in the computer with their structure, and you can design one antibody simultaneously to have a good binding affinity to all 35. But these are millions and millions and millions of atoms in your computer. And so for that, you need very strong multi-state design algorithms. And so we have developed some specific algorithms to tackle that task, and then showed that with those algorithms, we can indeed design antibodies. And so here we have one of those protein neutralizing HIV antibodies. So the ultimate goal here is a vaccine for HIV. To get a vaccine, we need to know the best antibodies. And this is about finding the best human antibodies that are broad against many different strains of HIV. Here we see a co-crystal structure of one such antibody. That's an antibody PG9, came out of a patient, bound to a mimic of the surface protein in orange or in salmon up here. The viral protein has these sugar molecules on the surface protecting it. That's why many humans will not make good antibodies against HIV, because our immune system matures antibodies that don't bind to points of these proteins that are constant, that cannot be changed. But sometimes it happens, and this antibody came out of a patient, and it turned out to be a protein neutralizing antibody. So it winds, this long CD03 winds in between the glycans and binds to a position of this protein that cannot easily be changed without the virus losing its function. This antibody came out of a patient, and here's a representative set of 30, 35 or so strains. This is binding data, this is neutralization data. Black box is PG9. Green means it's, so the lower the number, the better. The numbers are hard to reach. That's the concentration needed to neutralize. And you see it neutralizes about half of this representative panel. But this came out of a patient. The patient wasn't infected with all of these 30, obviously, right? It was just infected with one and then possibly mutations thereof. So we can put this in the computer and redesign this loop and ask what's the best sequence to neutralize them all. And if you do that, we tested, in this case, 4 different variants of this. If you do that, we found this one variant, this one mutant here, which is about twice as broad, neutralizes about 80% of the panel, and it's about 4-fold as potent. So you need 4 times less of the antibody to neutralize. And this knowledge is important if you want to engineer vaccines, because what you now can do is you can do something with Rosetta computationally that's called epitope-focused vaccine design, instead of using this entire protein as vaccine, right? This is what happens with SARS right now, right? We use a large portion of the surface protein of SARS as vaccine. And that works for, luckily, it works for coronavirus pretty well. But for something like HIV, this wouldn't work because the immune system makes too many antibodies to regions that are not important. But if we can computationally cut out this little region where these good antibodies bind, design a small protein that just displays that region and use this as a vaccine, the immune system will be forced to make antibodies that target that region. And so there are obviously long stories here that we won't have time to cover, but let me show you how this looks like. So this is, we switch viruses. This is Ebola, Marburg viruses. They belong to the same class. They have this surface protein here. There's the viral membrane, then there's an alpha helix that comes out, and then they have this long, it's a trimeric protein. They have these large domains out here. And at the time, a few years back, when we had these Ebola outbreaks in Africa, you know, my colleague here in the vaccine center, Jim Crow, they got blood from patients who survived this Ebola infection and sequenced with next generation sequencing their antibody repertoire to find antibodies that neutralize Ebola. And so BDBV223 is one of those antibodies, and it binds to this mPOR region. So the human immune system will make few antibodies that bind here, because here's the membrane, there's this huge domain. It's very difficult for the antibody just to physically get there. But this was a very good and very broad antibody. And so how can we design a vaccine that is focused on these type of antibodies? And so we took this little alpha helix and we looked for proteins that can stabilize this helix in its crystallographic conformation. We put the sequence on for the mPOR region, the residues that are critical for interacting with this antibody. And then you get a very small protein now that binds very tightly to this antibody that you want to elicit. Now, such a small protein is not a good vaccine because it doesn't look like a threat for the human immune system. And so what we did is, in this particular case, we use a Rosetta engineered platform that these are two proteins, they self-assemble into these little soccer balls. And on the terminus of one of those proteins, we added our little green protein, our immunogen. And so when it assembles, it displays 60 copies of this little protein on its surface. So that looks much more like a virus. And so you have wonderful EM images of this little soccer ball. And if you inject that into rabbits, then you indeed see that these rabbits start making antibodies that target this particular region of the glycoprotein. And so you have sort of a pipeline how to engineer the best human antibodies and then use these antibodies to reverse engineer the vaccines that elicit these antibodies. And this approach is very exciting. It's much more complicated than I had time to explain, so it will take a little bit of time, but I'm pretty optimistic that we're gonna have vaccines against viruses such as the HI virus or influenza virus that are protein neutralizing and protecting and can be given to kids and protect them for maybe not their life, but for 10 years, and then you need a booster shot or something like that. With that, I would like to thank the people who did the lab. This is obviously who did the work. This is obviously a photo of the lab out of Corona times here, the lab meeting. And obviously, I also want to acknowledge our experimental collaborators. None of the computational work is worthwhile without people who test your crazy ideas, and the funding agencies for continuing to support our research. And with that, I think I'm closing in time, and we have 10 minutes also for questions.
Okay, thank you very much. There are lots of topics, very interesting ones, very stimulating figures. So let's open the floor for questions. Any questions from the audience? You can speak up.
Yeah, speak up or type it in the chat.
Let me start with one. Very, very interesting talk, and thank you very much. So I have I have a couple questions, but I'll ask one, then wait if anyone wants to ask. Then we can touch bases again. So these antibody designs are amazing, or optimizing the antibodies to make sure that now they recognize a range of different strains. And that's a wonderful way of thinking about it. Where do you see the balance of like redesigning one antibody to recognize 20 different strains versus breaking up and making it two different antibodies with slightly different things to have like better coverage? Because at some point you're going to hit the issue of like the neutralization is affected for some of these strains to just compensate for covering more of these.
Yeah, I think that's a great point. Let me first say, I don't know. Secondly, we are developing computer algorithms that ask this question: what is the smallest possible antibody cocktail that neutralizes all known variants and all expected escape mutations? Sort of trying to play this game and predict what mutations you might see in the future in a virus and designing your antibody cocktail so that they can't escape. That's answer one, but that's still, research that's very, very basic science research. The second answer is, for the HIV vaccine, we do not believe that we will have one vaccine that elicits one type of antibody that will partly neutralize the virus. There are actually 3 or 4 good places on the surface of GP120, the surface protein of HIV, for partly neutralizing antibodies to bind. So the strategy here is to hit HIV with 3 vaccines that elicit 3 partly neutralizing antibodies targeted to 3 different regions on the surface of GP120, so that a single mutation in one of those regions will not be effective in saving the virus. And that the chance that the virus makes 3 mutations in the 3 regions simultaneously, stays, infective and escapes the antibodies, this chance is minimized. Similar to how you're giving a cocktail of 3 drugs right now to HIV patients to bring the viral load down, right? And that's the same concept here. So what actually is happening right now is the other thing I should say is it will not be a single vaccine. It will actually be a sequence of 2 or 3 or 4 vaccines that you have to take for each of those viruses. So probably you get a mixture of 3 initial vaccines and then 3 booster vaccines and then another 3 booster vaccines. And what's currently happening is these initial vaccines that sort of try to identify the right precursor antibodies and mature them at the first mutations to make them broad. These are in clinical trials and tested. And once we know what they do, and that's not, I shouldn't say we, this is there are many, many labs involved in this. This is a huge undertaking, right? This is by no means, I want to suggest this, that this is, we do a tiny fraction of this work. But once we know the antibody response to this initial vaccines, there are already versions for the booster shots that we can then test. These clinical trials will go on for several years, and probably there will be complications and yada, yada, yada. But ultimately, I think the question that you asked is a very important one. And we attack that question at the moment, I believe, by using 3 vaccines that target 3 different epitopes with 3 different very specific broad antibodies, trying to basically dead end the virus. There might be a combination of 3 mutations or 6 or whatnot that rescues the virus, but just basically ruling out the chance that the virus can find that combination.
Thank you.
Hi, Jens. I have a question about the ligand docking and approaches you're taking. So some of these machine learning approaches, you don't get like underlying knowledge of what's driving the descriptor, or which descriptor is driving docking and stuff. Can you comment on that issue and how we can maybe get some information that's useful for, let's say, a medicinal chemist or others to follow up on?
Yeah, we actually just published a paper on interpretable AI and how to back translate some of this information into a pharmacophore model. I forgot the citation, but it's
I can look for it. Yeah.
Ben Brown is the first author, and I think this is why I'm a big fan of the old style descriptors, because they are individual geometries, and that means if I can run earlier, I always called it a sensitivity analysis, but basically a neural network is nothing but a function, right? You have some input and you have some output. And what you can always do numerically is you can compute a partial derivative of your output with respect to each of the inputs, or even combinations thereof. And so if your inputs are specific structural features of your molecule, you can compute the derivative, how much does wiggling that feature changes the output, and that tells you something about the importance of this feature for the prediction. And so we back translate that into sort of a color-coded pharmacophore map that helps you to get some idea. This is very, we put these always in the discussion section of the paper and say, oh, we see something and it makes some sense. It's very difficult to do it the other way around, to sort of say, you use this as a design principle, because this is very noisy. And obviously, the partial derivative is in the first place a pretty, it's a delta y over delta x, right? So it's a linear thing. And then the neural network inherently has very complex nonlinear relations where multiple inputs need to be combined. And so mapping it back into a simple pharmacophore is not always trivial, but it gives some insight in that respect. But I agree, this is something where interpretable AI, sort of learning how to understand why the neural network comes to the conclusion it comes to, is a very exciting field of research, also for drug discovery, no questions. It's also some of these more recent approaches is like, we have this explosion of data and you just put all the data in without thinking too much about it. And I'm very skeptical for these 2 reasons. The first reason is I'm concerned about generalizability. I believe I can get better models that generalize better if I think smart, if I have smart descriptors and not just, and then interpretability. If I have smart descriptors, I can interpret what they mean for the model later on. While if I just dump the whole data, it's sometimes difficult to understand what's going on.
Yeah. Okay, thanks.
I have a quick question about this ligand drug design. So, from your experience, to make the computational protein model to work for useful for drug design, how accurate the models should be? Do you need to do you have some like RMST value or some sense about the accuracy you need?
1.5 angstrom.
1.5. Oh, that's a that's very high.
That's usually what I tell the students, right? Obviously it varies, right? And so, but in the most cases where when protein engineering worked, the design protein structure is within 1.5 or 1.5 to 2 angstrom. And also for ligand docking and design, I usually have this cutoff of 2 angstrom. But the problem is whenever you are larger than 1.5 angstrom, you lose the energy function is not good enough to see the energetic minimum. Sometimes it fails even if you are at 1.5 angstrom, but often it works if you are close enough in geometry. And you know, the 1.5 angstrom is not written in stone, but so over my experience, you need to be pretty close.
I see.
Yeah. The good thing is that the energy, you usually see that because the energy funnels are pretty tight. And so you usually, if you design a protein or a small molecule, you recognize, at least if Rosetta thinks there is an energy funnel, doesn't mean that there is one in reality, but at least it means, you know, we never test something in the lab. These small proteins, they all are designed, and then we do forward folding. And only if Rosetta folds them into the correct structure and not in alternates, only then we go to the lab and make them, right? And similar with yeah.
I have a question.
Yeah.
Yeah. We're having trouble designing a Syk kinase inhibitor that's membrane permeable. We have great success in making something that's really extraordinarily specific for Syk, and that is also very high affinity and exhibits every beautiful property you'd like in a drug except getting it across the membrane. And it doesn't look like it ought to be membrane impermeable, but it is. So do you have any way of using machine learning to guide someone like myself in converting an otherwise not too accessible drug into one that crosses a membrane?
I think that's a great question. I don't have anything readily available. We have some machine learning models that predict sort of DMPK parameters, but the last time we visited that topic, the data sets were still pretty small to train good models, and so you would need a very good data set on membrane permeability to train something like this. And I didn't look at this for probably 10 years, so I'm not sure what data sets are out there. In principle, you can train a model if the data are out there to train it, but our models are 10 years old and probably not that good at this point. But yeah, it's also, I mean, I'm part of a ton of drug discovery projects, obviously, here in academia, but also in collaboration with companies. And it's sort of, early on, we put effort into building such models, and later on the companies tell me, and the collaborators tell me, at this stage, when you have a molecule that's really active on your target and your assays work and everything, you don't trust the DMPK computational models anyway. You actually want to, test this experimentally. And so that has less been of a request of people to do these type of predictions. But yeah, I think they're super important. I agree with you.
Okay. All right. I think it's now 1:30. Yeah, let's thank Professor Meiler again. Thank you very much for your wonderful talk. Hopefully we can see you physically in a meeting sometime soon.
Yes,
you
yes,
very much.
that would be nice. Then I can get the dinner and a glass of wine
Yeah,
for
sure.
my efforts. That would be very appropriate. And then also, yeah, thanks so much for the invitation. Sorry for flying at 10,000 feet, but I think it would be good for this crowd to sort of give a little bit of an overview where what's happening. Okay,
Thank
thanks
you
so
very
much.
much.
Thank you very much.